Hi, I am Sharon Cantor, and I study breast and ovarian cancer, specifically the kind of cancer that is inherited. Today I will tell you about a new model for an old field, discovering the gap in chemotherapy. When you learn you have cancer, you want the doctor to say, we have a drug that will kill the tumor and only the tumor, but you are going to be perfectly fine. This is called precision medicine. It works when we know the specific genetic information for your individual cancer, and when we have drugs to target the specific genetic information. People are really excited about precision medicine, especially when it comes to treating hereditary breast and ovarian cancers. These cancers are thought to be vulnerable to drugs that cut the tumor DNA in half like a chainsaw. Indeed, this concept has bolstered a billion-dollar industry generating drugs thought to kill tumors by breaking the tumor DNA in half. Despite all this drug and tumor knowledge, people are suffering. Sometimes chemotherapy does not work, or the therapy ultimately fails and the patients die. Therefore, something in our knowledge of cancer and the drugs we're using to treat cancer must be wrong. When we try to understand how biology works, we scientists construct stories. These stories have been handed down and sometimes have been held onto for decades. Because of this, one of the most dramatic things that can happen in the career of a scientist is to figure out when deeply held stories do not fully add up, do not fully explain the data. It is like Galileo, when he observed that the planets do not revolve around the Earth, but rather around the sun. He faced lots of pushback, challenges to his new understanding, but often when a story doesn't explain the data, this is a real opportunity to see the world in a new light. My Galileo moment came when our data did not support the age-old model that chemotherapy works by breaking the DNA in half. But before I tell you more about that, first a bit of a backstory. When I was a girl, I was shy, but I had to be bold because I had an older, stronger, very competitive brother, and I wanted to show him I could do all that he did and better. Flash forward to now, I am a scientist. I am working in a field that has long been dominated by men. Like growing up with my brother, I wanted to show them I was just as smart and just as bold as they appeared to be. So I never imagined that shy me could tell my colleagues in the entire daunting field of cancer genetics that the story of cancer therapy was wrong. I asked myself, could it be true? The long-standing model that chemotherapy destroys cancer cells by chopping their DNA in half is wrong? The chainsaw model appeared very well supported. Here's some of the evidence. After chemotherapy, some cancer cells can't fix their broken DNA. These cells die. Other cancer cells can fix their broken DNA after chemotherapy, and these cells go on to live, continuing the cancer. Thus, broken DNA was fixed and chemotherapy failed. Challenging this story told for decades, we found cancer cells that did not fit this model. The chemotherapy worked to kill the cancer cells even when they had the ability to fix broken DNA. Moreover, when we did an experiment with the latest cutting-edge device to look at the tumor DNA as it was bombarded with toxic chemotherapy, we were stunned to find little evidence that the DNA was cut in half. Instead, we observed the tumor DNA became filled with holes as it was attacked by the chemotherapy. So rather than a chainsaw, the chemo was acting more like a woodpecker, and the woodpecker holes accumulated through chemotherapy treatment. The cancer cell next became very sick, and like a dead tree, fell over and broke apart. At this later time, the tree looked like it had been chainsawed. Thus, it appeared that the old chainsaw model was based on dying or already dead cancer cells, suggesting that these woodpecker holes were central to selectively killing tumors. We found chemotherapy was most effective in tumors that already had holes in their DNA, and said another way, tumors already had fragile DNA were more likely to be killed by the chemotherapy. Even more telling, when cancers learned how to defend against chemotherapy and became chemo-resistant, we found that they filled the holes and their DNA became solid again. The concept that chemotherapy does not cut the DNA in half, but rather riddles the DNA with holes, tells a new story about how chemotherapy works. This new story also helps us to a new step in the treatment of cancer. Instead of determining whether a tumor has defects in fixing chainsaw DNA, we should determine if the tumor has fragile DNA with holes, because the holes in the tumor DNA is a marker that tells us whether the tumor will die with chemotherapy. This understanding will allow us to make drugs that maximize DNA holes and combine them with drugs that prevent tumors from making their DNA solid again and chemo-resistant. Together, these insights will ensure chemotherapy targets vulnerable tumors and fully succeeds in killing the tumor. And it adds another chapter in the book of cancer genetics where a long-held belief is overturned.